Welcome everyone. Today I'll be solving question number 41 of the 2023 GATE Biotechnology paper. And the question is that which of the following statements is or are true about induced pluripotent stem cells? The first option is that they can self-renew. The second option is that they require specific signals to maintain their stemness. The third option is that they cannot be genetically manipulated. And the fourth option is that they can form organoids in vitro. We will see what each of these options mean. But before we do that, let's first understand what is an induced pluripotent stem cell. Before we know that also, we need to know the different kinds of stem cells found in nature, right? So let's see that first. So stem cells can be of three types. They can be totipotent stem cells, like in the first case, pluripotent stem cells, or they can be multipotent stem cells. So a totipotent stem cell is a single cell ability to proliferate and create differentiated cells in an organism. And this can include extra embryonic tissue. That means totipotent stem cells can become a complete organism by itself, like in this case you see. Pluripotent stem cells, on the other hand, can form or differentiate into any cell type of the body, but not extra embryonic tissue. Pluripotent cells cannot form a complete organism by itself. Like in this case, they form ectoderm, mesoderm, and endoderm, which in turn form skin, muscle, and liver, and other cells like that. The last sort of stem cell is multipotent stem cell, which are capable of differentiating into multiple specialized cell types, which are present in a particular tissue or organ. Like in the case of blood stem cells that you find in bone marrow, like you can see here, they are capable of forming white blood cells or red blood cells by differentiating, but no other type of cells. They cannot form liver cells by themselves, right? So these are the different kind of stem cells found in nature. So the question states about the statements of, which are true in respect to induced pluripotent stem cells, right? So let's see what is the difference between normal pluripotent cells, which we get from the inner cell mass of a blastocyst, and induced pluripotent stem cells. So induced pluripotent stem cells are not derived from the inner cell mass of the blastocyst. They are derived from somatic cells, which are fully differentiated cells found in our body. Okay. And these cells are reprogrammed using viruses, like you can see here, with certain factors like SOX2, CMYC, OCT4, KLF4, etc. And these factors, when induced into these somatic cells, causes them to become of a pluripotent state. So we call them induced pluripotent stem cells. And these can be used for researches. So now that we know what pluripotent stem cells and induced pluripotent stem cells are, let's see the options again. So let's look at the first option, that they can self-renew. So self-renewal is a process by which stem cells divide to make more stem cells and to perpetuate the stem cell pool throughout life. So since induced pluripotent stem cells are still pluripotent stem cells, they have within themselves the self-renewal capability. So the first option should be correct. The second is that they require specific signals to maintain their stemness. So what stemness means in this case is to the molecular processes underlying the fundamental stem cell properties, like self-renewal and differentiation, etc. So like I said, there are certain factors which need to be induced in somatic cells to make them pluripotent, induced pluripotent stem cells, right? So they do require specific signals to maintain their stemness. So the second option should also be correct. They cannot be genetically manipulated. That is the third option. But there is nothing stopping as such induced pluripotent stem cells from being genetically manipulated. So this is not a correct answer. And the last option is that they can form organoids in vitro. So what are organoids? Organoids are very simple tissue engineered cell-based in vitro models, right? Which are made using an aggregation of different cell types found in an organ that you will find inside an organism, right? And they can replicate many aspects of the complex functional and structural properties of the said tissue. And these are very helpful in researches. So it is also true that induced pluripotent stem cells, which can differentiate into cells of a particular lineage, can help form organoids, which are similar to certain organs, right? So the correct answers in this case are A, B, and D. C is not a correct answer. So that's it for this question. I hope this was easy to understand. Thank you so much for listening.